Hello bio students, this video is about viruses, and in this video we are just focusing on the learning outcomes. In other words, what you need to know for exams. All right, the first learning outcome is about what viruses have in common. What structural features do they have in common? First, they're all very small, smaller than bacteria. Second, they all have genetic material composed of either DNA or RNA. Third, they have what's called a capsid, which is made out of protein, and that encloses the genetic material. Fourth, they do not have cytoplasm, one reason they're not considered to be alive. And fifth, they have very few or no enzymes, so they don't have the ability to carry out the metabolic reactions that living things have to carry out to live. Another reason they're not considered alive. Despite the similarities they have, there's a lot of diversity in the structure of viruses. You can see in this sort of chart here, there's diversity in the nucleic acids that they contain. They can either have DNA or RNA as their nucleic acids. And the DNA or RNA can either be single-stranded, so that's represented by the two S's there, ssRNA, for example, or ssDNA, or it can be double-stranded. And notice that this is quite different than what we've talked about before for genetic material in eukaryotic cells, where RNA is always single-stranded and DNA is always double-stranded. There's more flexibility in what's possible. The differences in genetic material do result in sort of differences in how the viruses function to replicate themselves when they infect a host cell. The other major difference they have is whether or not they're enveloped or not. And when we say enveloped, we're talking about a plasma membrane. So some viruses are not enveloped, they're naked, you can call them naked. They don't have a plasma membrane around them, whereas some do. And the plasma membrane comes from the host cell where they were formed. Examples of viruses that you need to know about are bacteriophage, bacteriophage lambda, specifically coronaviruses, and HIV. So just looking at those in more detail, you should be able to sort of recognize bacteriophage lambda for sure from an image. It has a very distinct structure. Its genetic material is double-stranded DNA, and so it will inject that DNA, which then within the host cell has to be transcribed to mRNA, which is then used to make viral proteins. It has a very complex capsid shape, and it is not enveloped. Coronavirus and HIV are enveloped. They both use single-stranded RNA as their genetic material. And they both are quite similar in how they look, in that they have this envelope with spikes on it, proteins on it that allow it to dock to a host cell. They are different in their caspids. The capsids, sorry I said that wrong, the coronavirus one is helical. It's sort of in that diagram, the red helical structure right within the plasma membrane. And the HIV capsid is in the middle, and it's more like a cone shaped. It's that yellow structure in the diagram. Looking at the life cycle of a virus, you need to know specifically the life cycle of bacteriophage lambda. You do not need to know about the life cycle of other viruses, but this pattern holds true for a lot of viruses, so it's not only true for bacteriophage. So bacteriophage can follow a lytic cycle, and what that is the process by which they are able to create copies of themselves, which then emerge from a host cell. So what they do is they first attach to a host cell, they inject their genetic material, their DNA in this case. The DNA is then used to make viral proteins, and those proteins are assembled to make new viruses within the host cell, which then burst out. That's called lysis, killing the host cell and releasing the bacteriophages into the environment where they can then infect other host cells. The lysogenic cycle is also happening in bacteriophage lambda. And it's what happens sort of when times are good. That's the way I think of it. So just like the lytic cycle, the bacteriophage lambda will inject its viral DNA into the host cell, but then that viral DNA becomes incorporated into the bacterial genome. So it first becomes circular within the host cell. That's necessary for it to incorporate into the bacterial genome, and then it becomes part of the bacterial chromosome, but it basically becomes deactivated. So it doesn't actually, it's not used to create any proteins, any viral proteins, it's just there within the cell. And whenever the bacteria then divide, in bacteria it's called binary fission, they make a copy of their DNA, and in doing so they also copy the viral DNA because it is inserted in their genome. And so in this way the viral DNA gets replicated even though it's not used to create new viruses. In this cycle, the inactive viral DNA that's part of the bacterial DNA is called a prophage. Now, the virus can become active and switch to the lytic cycle. And so that often will happen when times are bad. For example, if the bacteria are exposed to UV light or a sudden change in their chemical environment. And when that happens, the viral DNA will be cut out, will remove itself from the bacterial genome, and then it will be used to make viral proteins, which are assembled into viruses, which then break out of the cell again. So that's the lytic cycle. Infected bacteria are fine if the virus is in the lysogenic cycle, but if the virus switches to the lytic cycle, then those bacterial cells will burst, and the viruses will escape, will get out essentially, and be able to infect other bacteria. So that's sort of the basics of viral structure and their life cycle. This next bit here is on, well, where did they come from? The idea is that, well, not the idea is, we know that viruses need host cells, right? That's sort of how we define a virus. It can't replicate by itself. So if it needs a host cell, then it would make sense that cells evolved first. The other thing we know about viruses is they use the same genome, not the same genome, but the same genetic code, the same nucleic acids genetic code as cells. And so that further supports the idea that they were once cells, that they evolved from cells because they use the same genetic code. Today, viruses are essentially like a parasite. They cannot exist on their own. We sometimes say they are an obligate parasite, so that's their only way of existing. There are a couple of theories about how they evolved. One is that they evolved from fully functioning independent cells that regressed or that lost function. For example, maybe a cell lost its ability to copy its genetic material, to replicate its genetic material. And then those regressed cells essentially became parasites of other cells, using other cells to do the things they couldn't do. And so they evolved into viruses. And maybe that was a more successful way of replicating. And so it was selected for, and those viruses that did that increased over time. The other idea is that maybe viruses evolved in a progressive way. So maybe there were bits of genetic material in a cell that gained function. They gained the ability to infect other cells and use those cells as a host. There are these little bits of DNA that exist in cells today, like in bacteria. There's sometimes these separate little bits of DNA, separate from the main genome, called plasmids. And so it's hypothesized maybe these kinds of extra bits of DNA gained the ability by acquiring these sort of proteins around them to infect other cells and become viruses. So those are two hypotheses, but they both are suggesting that viruses evolved from cells. If viruses evolved from cells multiple times, then why are they so similar? So if there are these multiple evolutions of viruses from different cell types, wouldn't you expect them to be different? The idea to explain why they are so similar, why they have these similarities, is because they share the same kind of niche. All viruses are obligate parasites. And so to be an obligate parasite, you have to evolve certain abilities, like being able to dock to a host cell, being able to inject your DNA, being able to make use of the host cell's molecular machinery to create your own proteins and so on. And so the idea is that even though they may have evolved independently multiple times, viruses, since they share an ecological niche that they evolved to, they came to, over time, all these different viruses came to look very similar. So that's called convergent evolution. And that's where we have many different organisms that look structurally sort of superficially similar because they evolved to the same ecological niche. You can see in the diagram there a dolphin and a fish. They look similar, but a dolphin is a mammal and a fish is a fish, or a shark is a fish. And they acquired those similarities separately. It's not like they looked similar because their common ancestor looked similar. They look similar because they evolved to the same environment, and so they look similar today, even though they evolved independently of each other. Divergent evolution is sort of the opposite, where organisms that share a common ancestor come to look quite different because they have different ecological niches. Darwin's finches there are a good example of that, where their beaks vary to match the kind of food that they eat. Viruses evolve very rapidly. That makes it very hard to protect ourselves from them. We're always sort of in an arms race. There's always an arms race between our immune systems and viruses, and today between vaccine makers and viruses. So why can they evolve so rapidly? You need to be able to give some reasons, and you need to be able to give examples in influenza and HIV. So why do they evolve so rapidly? One, their generation time is very short. So us humans, we live a fairly long life. It takes at least 25 years or so on average before we're producing children. So it's a long generation time. Viruses, it's less than an hour, so much quicker. Mutation rates are high. In us, for example, comparing to humans again, when our cells divide, the DNA is replicated very reliably, which is important because if not, we would end up with all sorts of problems with our DNA when we grow and develop. In viruses, they don't have the same proofreading mechanisms when their DNA or RNA is replicated. It's especially true for RNA. And so there's lots of errors when RNA is replicated in viruses. And you might think that's bad, but it also provides a source of new variation. And so if generation times are short and there's lots of mutation, chances are every once in a while you'll get a mutation that's good and provide some benefit and protection against immune system or a vaccine. That's why the flu every year is a little bit different because of this. And viruses are always exposed to a lot of natural selection. It means there's all these things trying to kill them all the time, specifically immune systems, right? So our immune systems are always trying to kill viruses, and well, now vaccines are also we're using vaccines as well, which is really just exploiting immune systems to destroy viruses. So they're under this pressure all the time, and therefore it's more likely that they will evolve quickly because the ones that are being killed will disappear, and the few that are left over that have been successful with mutations that give them some benefits will be able to survive and replicate themselves and increase over time. So the examples, influenza. Influenza uses ssRNA. It's negative sense RNA, which means it has to be a complementary strand of that RNA has to be produced first before it can be used to make proteins, and that leads to a lot of mutations. It also has more than one RNA strand. It has 8, and so they can be combined in different ways to make new strains. And if you're infected by more than one virus with different strains at the same time, those different strands can end up in the same host cell and produce a new combination of RNA of those 8 molecules and make a new strain. That's another reason they mutate quickly. Just a note, you probably heard like H1N1, H3N2 for the strains of the influenza virus. Those are named after 2 of their proteins, the ones that are on the surface, that end up on the surface of their envelope, their membrane, their outer membrane, and the H stands for hemagglutinin and the N stands for neuraminidase, and they allow the influenza virus to bind to host cells. So you can imagine if those proteins change, they can develop a new way to bind and to infect host cells. HIV has the highest known mutation rate of any virus. It is a retrovirus. You should be aware of that. You don't need to know about it in detail. Basically, it takes its RNA, which is its genetic material, and then it uses an enzyme which it has within it. So it's a virus that does contain an enzyme called reverse transcriptase, which creates DNA. And then that DNA is used within the host cell to create an mRNA, which is used to create viral proteins. The process of using reverse transcriptase leads to a lot of errors. So again, a high mutation rate. There's also, this is a confusing point, but there's this enzyme found in host cells that is part of natural metabolism called cytidine deaminase. But it actually, when the viral DNA is there, it changes that DNA, creating mutations in it. Normal enzyme, but it causes problems in the presence of HIV. And so the HIV actually evolves within an organism, within a host, after infecting the host. And so that allows it to exist within that host for a long time, evade the immune system, and essentially also evade drugs, like become resistant to drugs, and ultimately lead to chronic infections. We have very good drugs for HIV now, but there's not a cure. So you have to take the drugs your whole life, and the virus sort of is able to stay within your body forever and exist, partially because of this high mutation rate.